The Shroud of Turin is this amazing relic. At bottom, it's a piece of cloth. It shows a very faint sort of image of a crucified man. And the crucified man calls us to remember Jesus' death and resurrection.
In 2015, inside an ultra-clean genetics lab at the University of Padua, Professor Gianni Barcaccia and his team cracked open something that was never supposed to be found. Not a painting, not a pigment, not a forgery caught in the act. A biological code embedded in the fibers of the cloth millions believe wrapped the body of Jesus Christ after the crucifixion. For centuries, the debate had been simple. Either the shroud was a medieval fake or it was real.
The Shroud of Turin is a burial cloth that's imprinted with what many believe is the actual image of Jesus after his crucifixion.
DNA was supposed to settle it once and for all. European DNA would mean forgery. Middle Eastern DNA would mean authenticity. But when Barcaccia's computers finished decoding millions of nucleotide sequences and the final genetic map hit the screen, his team just stood there in silence. Because the secret code didn't point to one person, one country, or one continent. It pointed everywhere. And what it revealed was a journey so vast, so precise, and so impossible to fake that it collapsed every existing theory, all at once. Here's what they found. The face you're not meant to see. To understand why that DNA shattered every existing theory, we need to go back to the night the shroud first revealed what it was hiding. May 28, 1898. Turin, Italy. A lawyer and amateur photographer named Secondo Pia was granted rare permission by King Umberto I to photograph the relic during a public exhibition. Pia hauled a camera nearly the size of a suitcase onto scaffolding inside the cathedral. Using magnesium flashes to light the dark interior, he exposed two large glass plates. Late that night, alone in his dark room, lit only by the dim glow of a red safety lamp, Pia lowered one plate into the developing chemicals. As the image materialized, his hands started shaking. He nearly dropped the glass. What was forming before his eyes wasn't supposed to exist. On the photographic negative, where light should become dark and dark should become light, a sharp, high-contrast face appeared. Deep-set eyes gently closed, a broken nose. Mustache and forked beard, bruising along the right cheek. For the first time in history, the figure hidden inside the linen wasn't a faint shadow. It was clearly a man, calm, dignified, yet marked by extreme suffering. Now, here's the catch. Any normal image behaves predictably as a negative. Light turns dark, dark turns light, and the result looks distorted and unnatural. But the shroud broke every rule. Photographed as a negative, it appeared as a true photograph of a real human being. What the naked eye sees on the cloth is already a negative. Who in the Middle Ages understood photography 800 years before the camera existed? No one. The human eye cannot view the world in negative, let alone reproduce it with precise control over light and shadow. No artist, no scholar alive at the time possessed the knowledge to even conceive of such a thing, let alone execute it flawlessly on a piece of linen. This was the first crack in the wall of skepticism. The shroud didn't behave like a painting. It behaved like a photographic plate, capturing a single instant in time. And that realization sent a tremor through every discipline that would later touch this cloth. The DNA that shouldn't exist. In the 21st century, speculation finally gave way to something much harder to argue with: genetics, high-energy physics, and forensic molecular analysis. Scientists stopped treating the shroud as a holy relic and started treating it like a crime scene. A biological data vault quietly preserving 2,000 years of history. In 2015, Professor Barcaccia's team of geneticists and biologists was granted unprecedented access to the relic. Their goal wasn't to find the DNA of God. Science has no model for what such a trace would look like. They wanted to reconstruct the life story of the cloth itself, where it had traveled, who had touched it, whose hands had held it across the centuries. Using sterile micro-vacuum devices equipped with ultrafine filters, they collected microscopic dust and organic fragments from deep between the warp and weft threads, spaces where ancient material can remain sealed for thousands of years. And get this, they focused specifically on mitochondrial DNA and for a critical reason. Unlike nuclear DNA, mitochondrial DNA exists in hundreds of copies per cell. It's passed down exclusively through the maternal line, and it survives far longer in ancient material. It's essentially a biological GPS, a powerful, reliable marker of geographic origin and human migration. If any genetic information had survived inside those fibers across 2 millennia, this was the method that would find it. For weeks, computers ran non-stop, decoding millions of nucleotide sequences and comparing them against global genomic databases representing populations from around the world. Then the final maps appeared on screen. The lab went quiet. Nobody spoke. Barcaccia's team was staring at data that defied every prediction they had made. This was not the genetic signature of a single individual. It was something far more complex, something no one had anticipated. The findings, published in the highly respected journal Scientific Reports, detonated across the scientific community. Here's what they expected. If the Shroud were a medieval forgery created in France, European DNA should have overwhelmingly dominated. If it were an authentic relic from Jerusalem, Middle Eastern DNA should have dominated. Neither happened. First hit, the Middle East. Haplogroups commonly associated with the Druze, a tight-knit, historically isolated community in Israel, Jordan, Lebanon, and Syria, whose genetics have barely changed in millennia. That alone was significant. A powerful marker of geographic origin, strong evidence of a direct Middle Eastern connection dating back centuries. But then came the second hit. Western Europe, haplogroups, U5b and H1 through H3, consistent with centuries of European handling. The Poor Clare nuns who repaired it, the House of Savoy who owned it, countless pilgrims who venerated it across the centuries. Okay, that tracked. You could explain that. But here's where it gets wild. Third hit, North and East Africa. Haplogroup L3, pointing toward Egypt and Ethiopia. That was unexpected, deeply fascinating. It suggested direct contact with some of the earliest Christian communities on the African continent. The room shifted. Fourth hit, South Asia. Haplogroups M39, M56, and R8, genetic markers typical of the Indian subcontinent. Now the team was staring at each other, and then the fifth hit landed, the one nobody could process. East Asia, haplogroups D4 and G2A, commonly associated with China and the surrounding region. China, India, Africa, the Middle East, all encoded into a single ancient cloth. Let that land for a second, because the question now wasn't just what the DNA revealed, it was how it got there. And if a forger didn't put it there, who did? A world written on cloth. Now, pay close attention to this. No medieval craftsman working in a French abbey around 1350 could have collected genetic material from China, India, and East Africa. Globalization didn't exist. Long-distance travel occurred, but not on a scale vast enough to leave such widespread genetic signatures on one piece of fabric. Nor could such a person have intentionally planted those traces in a way that would fool genetic scientists 600 years later. The DNA found on the Shroud is not random contamination. It is a collective biological memory. When Barcaccia mapped the data geographically, the mystery deepened even further. The distribution of genetic markers aligned with remarkable precision along a specific ancient route, a path scholars had long dismissed as legend. According to early Byzantine, Syrian, and Arabic sources, the Shroud was folded so only the face was visible, a form called the tetradiplon, and displayed inside a special frame. This relic didn't stay in one place, and it certainly didn't begin in Europe. Its journey started in Jerusalem. From there it moved to Edessa in the 2nd century, where it remained hidden within the city walls for centuries. Edessa sat at one of the most important crossroads of the ancient world, the Great Silk Road. Caravans from China loaded with silk, merchants from India carrying spices, pilgrims from every corner of the known world. Many of them came to venerate the image believed to protect Edessa. They stood close, they kissed its casing, they touched the cloth, and with every encounter, microscopic traces settled onto the fabric, layer by layer, century after century. The DNA of the entire ancient world, accumulating on its surface like invisible dust. Here's the catch. This wasn't contamination. This was documentation. The journey didn't end at Edessa. From there to Constantinople in 944 AD, the greatest megacity of the ancient world, where people of every race and culture converged. Then, in 1204, during the Fourth Crusade, the city was violently looted. The Shroud disappeared. It resurfaced in Athens around 1205, passed through French knights, and finally appeared in France around 1353 in the possession of Knight Geoffrey de Charny. And it is here that the genetic evidence delivers a devastating blow to the medieval forgery theory. The DNA on the Shroud is not random noise. It is a geographic fingerprint that no forger could have manufactured. Dust from sandals, robes, and hands left behind by countless people who stood before this face over many centuries. A biological trail stretching across 2,000 years of human contact. And if the DNA was this precise, this globally mapped, this impossible to fake, what did the pollen say? Because the botanists found something the geneticists couldn't explain. Subscribe now so you don't miss where this ends, because the blood evidence is about to make everything else look like a warm-up. Pollen, blood, and a crown of thorns. The genetics were only half the secret code. The other half was written in plants, and it pointed straight to a crime scene. Professor Avinoam Danin, a renowned Israeli botanist from the Hebrew University of Jerusalem, had spent decades cataloging the plant life of the Levant. He knew its pollen the way a detective knows a fingerprint. When he began cross-referencing the species trapped inside the Shroud's linen fibers, the room around him stopped mattering. Swiss forensic scientist Max Frey had reached the same place independently, both men working from the same invisible evidence, arriving at the same conclusion. 58 different plant species identified, cataloged, and mapped. 17 native to Europe, expected, given centuries of European handling. But the majority came from elsewhere, the Middle East, Turkey, the Anatolian steppe, perfectly matching the ancient route through Edessa and Constantinople. And get this, scientists identified plants that grow nowhere else on Earth, only within a narrow corridor between Jerusalem and Jericho. One species dominated, Gundelia tournefortii, a thorny desert thistle. When Danin saw the concentration pattern, he stopped, said nothing for a moment, because the pollen wasn't spread randomly across the cloth. It was saturated around the head and shoulders, nearly half of all samples, clustered exactly where a crown would sit. The crown of thorns. Gundelia tournefortii, with its long needle-like spines, is exactly the plant Roman soldiers could have twisted into a mock crown. It blooms near Jerusalem in early spring, precisely during Passover. Here's the part they never put in the headlines. No medieval forger working in France could have obtained pollen from plants native exclusively to Israel, let alone applied it invisibly at a microscopic level across a piece of cloth. A forger would have used paint. Pollen cannot be painted. It functions like an invisible seal from a crime scene, a geographic fingerprint that cannot be faked. A second botanical clue came from Zygophyllum dumosum, a plant endemic to a very restricted region, growing only in the Judean desert and parts of the Sinai Peninsula. Its pollen was also found in significant quantities. Then came the blood, and the blood told its own brutal story. For years, skeptics argued the reddish stains were nothing more than pigments. That claim collapsed in 2017. Professor Giulio Fanti at the University of Padua assembled a team of physicists and physicians from a hospital in Trieste. They examined the stains using transmission electron microscopy and Raman spectroscopy. At the nanoscale, the data came back. Fanti looked at the results, then looked again. Not pigment, blood. Human blood. Blood type AB, one of the rarest blood groups in the world. This same blood type appears repeatedly on ancient Christian relics, including the Sudarium of Oviedo. A cloth tradition says covered Christ's face at burial. But here's what stopped them cold. This was not the blood of a healthy person. Within it, scientists detected nanoparticles of creatinine and ferritin bound to hemoglobin. Such extreme concentrations occur under only one set of conditions: catastrophic fatal trauma, prolonged torture combined with dehydration, and massive muscle destruction. When muscle tissue is repeatedly damaged, a process known as rhabdomyolysis, creatinine floods the bloodstream in enormous amounts, and the blood preserved on the cloth recorded exactly that. This is not symbolism. It is a biochemical signature of agony. The man wrapped in this cloth was beaten to a physical state already incompatible with life before. Crucifixion even began. Evidence of over 100 blows, consistent with Roman scourging using the flagrum. Leather whips embedded with lead weights. And this detail still haunts researchers. The blood stayed red, which should be impossible for ancient blood. Normally, old blood darkens to brown or black within weeks, but scientific analysis revealed unusually high levels of bilirubin, a substance released by the liver during extreme stress and severe trauma. Bilirubin preserves the red color of blood. For centuries, the blood of a tortured man stays red. Not mysticism. Biochemistry. A painter can imitate the appearance of a wound. No human hand can reproduce the biochemical signature of extreme trauma and hypovolemic shock. Every molecule pointed to the same place and the same event. The test that got it wrong. But what about the one test that was supposed to prove it was all a fake? In 1988, 3 prestigious laboratories, Oxford, Zurich, and Arizona Carbon-14, dated the fabric to between 1260 and 1390, medieval, a forgery, case closed. The world accepted the verdict. Even the Catholic Church stepped back, referring to the shroud as an icon rather than a true relic. Here's the thing, the technology wasn't wrong, the sample was. 3 decades later, researchers discovered exactly where the critical mistake had been made. In 1988, a tiny piece of fabric no larger than a postage stamp was cut from the very edge of the shroud. That corner had been handled countless times. Then chemist Raymond Rogers of Los Alamos National Laboratory found something devastating. That corner had been expertly repaired during the Middle Ages. The Poor Clare nuns had reinforced the damaged edge by weaving new cotton threads into the original fabric, dyed with alizarin, and sealed with gum arabic to match the aged linen. Here's the kicker. The main body of the shroud contains no cotton. At all. It is made entirely of linen. The laboratories didn't date the shroud. They dated the patch. The most famous scientific test in the history of this relic, the one that closed the case for a generation, was run on the wrong material. In 2022, physicist Liberato De Caro at the Institute of Crystallography in Bari introduced a radically different approach, wide-angle X-ray scattering, or WAXS. Instead of analyzing contaminants, this method examines the aging of the linen cellulose itself at the atomic level. Over time, linen naturally degrades. The polymer chains of cellulose slowly break apart under exposure to radiation and temperature. This process functions as the material's internal clock. De Caro compared samples from the shroud with fabrics of known age, from Egyptian mummy wrappings dating back to 3000 BC to medieval textiles. When the aging curve appeared on his monitor, he called his colleagues over immediately. The shroud's its structure didn't match medieval fabrics at all. It was far older. And get this, it matched linen recovered from the fortress of Masada in Israel, dated between 55 and 74 AD 1st century, the time of Christ. In 1988, science dismissed the mystery. In 2022, it brought it back. But the question left standing, the one no test had come close to answering, wasn't about the age of the cloth. It was about what created the image, because that part has no explanation at all. The impossible image. But one mystery still towers above all the others, and modern physics cannot explain it. The image exists only on the outermost 200 nanometers of the fibers, hundreds of times thinner than a human hair. It is a chemical transformation caused by oxidation and dehydration, a kind of scorch left behind by an unknown source of energy. Scientists at ENEA, Italy's National Energy Agency, tried everything to recreate it. Nothing worked. Only a brief, intense pulse of vacuum ultraviolet radiation came close, technology that didn't exist in the ancient world. Think about what that means. It isn't that we can't explain how a medieval forger did it. We can't do it ourselves. To imprint the image across nearly 4 square meters of fabric, the body would have had to release a burst of energy lasting less than a billionth of a second. Powerful enough to mark the cloth, yet precise enough not to burn it. And get this. In 1976, NASA analysis revealed the image contains perfectly accurate 3-dimensional information. The intensity of the imprint corresponds exactly to the distance between the body and the cloth. Not approximately, exactly. This is something no artist, ancient or modern, has ever been able to replicate. But here's the detail that makes even the most hardened skeptics pause. Digital analysis identified shapes over the eyes consistent with coins, matching. Rare lepta minted under Pontius Pilate around 29 AD. These coins were only in circulation for a brief window of time in a specific region. The likelihood that a medieval forger could have known or reproduced this detail is effectively zero. And it doesn't stop there. The nails pass through the wrists, not the palms. Exactly as forensic science predicts for crucifixion. Every medieval painting gets this wrong, every single one. Artists showed nails through the palms because that's what they assumed. But the shroud shows the anatomically correct position. Even nerve responses are visible, including the retraction of the thumbs caused by nail damage to the median nerve, a detail no medieval artist could have known. Here's where it all lands. Layer by layer, everything converges on one city and one narrow window of time, Jerusalem, between 30 and 33 AD. Here is what no one can get around. A medieval forger would have needed to fake a photographic negative centuries before photography, embed pollen from plants found only between Jerusalem and Jericho, manufacture blood with trauma biomarkers undiscovered for 600 years, replicate three-dimensional encoding no modern lab can reproduce, and collect DNA from China, India, Africa, and the Middle East, all from a French abbey with a candle and a paintbrush. The Shroud of Turin is not a painting. It is a forensic record of a real human being, a biological archive, a silent witness to a moment that altered human history. The secret DNA sequence mapped across its fibers traces a 2,000-year journey that no forger could have written. Do you think science will ever explain what happened inside that tomb? Drop your answer in the comments and subscribe, because what we're covering next is even harder to explain.